A particulate graft, in combination with autogenous bone derived from a bone trap at time of surgery, will be used in combination to carry out the bone augmentation. The Inion biodegradable membrane system is used to prevent the downgrowth of soft tissue, connective and epithelial cells into the underlying bone during the healing period. The membrane and templates are packed in a sterile plastic blister. After opening the blister, the template is removed from the packaging. The membrane itself can then be found in compartment A of the blister pack. A small plastic bottle of plasticizer is also provided with the package. The entire contents of the bottle should be emptied onto the membrane in compartment A. This should then be left for 20 to 30 seconds. The particulate is then thoroughly mixed with autogenous bone derived from the bone trap. This mixture is then slightly moistened with saline solution. The prepared graft is condensed into a dispensing syringe. Excess moisture is then removed and the graft can be put to one side ready for use. The plasticizer has been allowed to diffuse in the membrane for about 10 minutes. It is then rinsed with sterile water and the membrane will then be ready for use. A template is shaped to an appropriate size to provide guidance for the surgeon to subsequently prepare the membrane. Using the template, the prepared membrane can now be accurately trimmed to a suitable size to cover the bone defect. The next stage is to unwrap the tack fixation kit and carefully open the plastic dispenser without dislodging the tacks. The sterile burr provided should be loaded into the contra-angled handpiece. A tack is then fastened to the tack applicator. The graft mixture, which was prepared earlier, is now used to augment the bone defect. The mixture is introduced using the delivery syringe. The mixture is moulded and condensed to ensure complete infill around the implants. The prepared and carefully shaped membrane can now be placed in position. A hole is now drilled through the membrane into the bone, and while keeping the membrane in position, the loaded tack applicator is then pressed firmly into the hole. Further tacks can then be placed as needed until the membrane is firmly stabilized. The surgical site is now closed using vicryl sutures and the suitably adjusted temporary denture refitted. The procedure is now complete.